Scripps research.
Hi everyone, my name is Larry Rodriguez. I'm a postdoc in the laboratory of John Yates at Scripps. And today I'm going to be talking about electrophysiology-informed single-cell proteomics. And to just provide a quick disclosure, some of our co-authors today were employed by NGB AI during the process of these experiments. So the first thing I'm going to talk about at ASMS is actually the electrophysiology side, if you're unfamiliar with it. So I do brain slice electrophysiology, which is what it sounds like. We have thin brain slices that I find under a microscope, under 40x, to look for individual neurons. And I'm recording the electrical activity of this individual neuron. And we do that because we know that neurons communicate through ion channels. And we know other proteins at the synaptic level control this output. So we have a high-functioning sort of electrophysiological system, but the downside is that it's ultra-low throughput. So from 8 a.m. to 5 p.m. of patching, I might get 2 data points, and you can imagine that this is very slow, which is a perfect application for single-cell proteomics, right? We could translate the signal that we're getting at the electrophysiology level to a molecular dictionary. And we're in the age where the instruments are providing a lot of content and information at ultra-low input. So the question I also want to ask is the one that I always ask in lab, and since I've been learning things, why is this so hard? So there have been publications in the past trying to incorporate this sort of idea, combining patch clamp and single-cell proteomics. But they've taken a 3P collection strategy where you assume electrophysiology will automatically give you good proteomics. And what I mean by the 3P strategy is you first start with a patch clamp experiment. So in this, you're actually having electrical contact with the intracellular side of the cell. You form a circuit, and you're able to pump current through this to be able to determine things like the size, the integrity, and the health of the cell, in addition to the kind of cell you have. The problem starts when you actually try to pull the cell out. So typically, you either jam pieces of the cell into the glass electrode that you're using to make these recordings, and in the process, you lose what we call the gigaseal, which allows these electrophysiology recordings. So you have no idea the size or the integrity of what you're collecting. And the last step is that you pretty much got to pray that you still have some of your sample, and at the single cell level, it's pretty bad because you might spend a week or two trying to collect some experiments, and if you're not getting any proteins, then you're just going to have to start at square one. So to formally present the problem, proteome recovery may not reflect the tissue electrophysiology of the neuron. So my first idea was to just do more electrophysiology, where rather than losing the seal when you're trying to pull it out, preserve it. So in theory, if you can keep the circuit alive during this process, you should be able to continue the electrophysiology recording while you're pulling the neuron out of the tissue. This will tell you, at the very least, the size and the integrity of the neuron. So when you hand it to the person running the mass spec or doing some of these experiments, you could say, this is a bigger cell, we're expecting this kind of proteomics, if that's actually what is going on. The problem is, I already told you that this is an ultra-low throughput sort of system, and doing additional electrophysiology experiments is impractical because it's really, really hard to even get a good recording and still lose the seal. So at the Yates lab, we provide solutions, and one of the solutions we came up with was a shotgun collection. So the goal here is now to use proteomics to fill in the gaps in electrophysiology. So what I mean is that we're actually collecting whatever we get during the patch clamp experiment. So like I said, sometimes we fail, a lot of times we fail in electrophysiology. And even if we are unable to get a recording, we're pulling the neuron out. So say you don't fail that experiment. You're able to do a typical electrophysiology recording. When you do the removal, there's basically 3 outcomes. You either lose the cell, like before. So you have the recording process, you lose the gigaseal, you can still see the cell, fine. This is a typical approach that's been done. Or, worst case scenario, you do the recording, and then you tear the neuron. So this will actually give you worse proteomics because you know that you don't have what you're expecting because you lost it and you can't see it. If you're lucky and good, then you can actually get to the end of this process where you've preserved the electrophysiology seal during the process, and you're able to record from the neuron as you're removing it just before you prep your sample. And what this looks like as an overview is that you're doing an electrophysiology experiment. We started with the cortex of rats and just try to patch. Because again, regardless of what happens electrophysiologically, we're going to be preparing the neuron. And we're doing a one-pot sample prep. So at the end of the collection, I take the recording electrode, which contains the neuron. I break it into a DDM-coated 384 well plate. And at the end of the day, I trypsinize and prepare my sample before it goes into the mass spec. So we ran these experiments on NGBAI's Vanquish Neo system on a 2-hour gradient with FAMES on an Astral in DIA mode. And our experiments were analyzed using Diane with MBR enabled and a 1% FDR at the protein level. So I just want to quickly show you what I mean when I'm trying to do these experiments. If it's hard to see, it's partially my fault. These are recordings from the image of what I see under the rig, but it's really hard to see. So for the neurons that have no electrophysiology, I just want to see something at the end of the tip. So I've highlighted it in red for one that has no electrophysiology. If I got some ePhys recordings, but I wasn't able to maintain the seal, then, as others have reported, you have the neuron here in the shank of the electrode. And if by some miracle I've been able to preserve the electrophysiology seal during these experiments, then, again, we have it at the end of the tip right here. So just a quick overview of the results. We have 12 neurons. And interestingly enough, you see roughly similarity protein ID-wise. But we have some promise because the Tor neurons that I said earlier are a kind of internal control of something that's not good, where we can't see something, is at the lower end of the protein IDs. So again, electrophysiology done at the tissue level will tell you what kind of cell type you have. Here is some electrophysiology jargon, Rio-based amplitude. But it doesn't take an electrophysiologist to see that these neuron responses are not the same. So again, at this level, it'll tell you what kind of cell you have. So when you combine these approaches, what does the tissue electrophysiology tell you about the proteome that you're recovering? If you just look at the numbers that were taken in the tissue, not much, because a measure of neuron size, capacitance, has no real correlation to what you're getting when you run this on the Astral. Neither does neuron integrity. So for membrane resistance, this is how well the membrane of your neuron is intact. But that's also not completely true, because some of these cells do have additional recording. These are just the recordings that I did in the tissue for these green ones. And again, it still kind of makes sense that regardless of the size, if you can't see the cell that you've collected, you probably don't have very much of it. So this might be a little confusing. Does tissue electrophysiology even matter if you're getting good protein IDs? And if you do a PCA plot of some of the proteomes, then you'll see imperfect separation, in that we do have a group of the electrophysiology none category, where they were just ripped out, clustering near one of these that has the gigacil preserved. So we got characterization. On the other hand, one of these cells, the one with the biggest protein IDs, clusters around those that have been well characterized in the tissue, but don't have much recordings after that. So do you really even need to do these experiments? Remember, yes, because it's biologically cell typing the neuron that you have. So let's flip the question then. Are protein IDs everything? So what I'm really asking now is this a quantity or quality type of thing that you're looking for? So if we take some of the representatives for each of these groups, the neuron with most IDs, number 1. The neuron with the most IDs with electrophysiology performed in the tissue, characterized, number 3. And my personal favorite, neuron number 4, which has, in my opinion, the best electrophysiology. What will that tell you? So for these experiments, I'm taking the proteomes and I'm running them through Syngo, which is a curated resource for gene ontologies related to the synapse. So it is curated by experts. No AI goes into this. And I'm running this under pretty stringent filtering. So we need multiple levels of molecular biology experiments, publications. And I'm just looking at the biological processes that come back significant at a Q value of less than 0.05. And we can see that, again, we're getting a lot of protein IDs that are fairly similar, and we're getting fairly similar gene ontologies as significant across most of these neurons. But when you start looking at some of these different groups that I was talking about, then it's not that important for patch-SCP, because the neuron with the most IDs, more than 2,400, neuron number 1, had no electrophysiology and is more towards the lower end of information biologically. Alternatively, the neuron with more electrophysiology and with a good number of protein IDs is more towards the bottom. So there's more biological information present in this neuron. And of course, my favorite neuron, number 4, had the best electrophysiological characterization and had the most number of significant biological processes. So this does suggest that it's a quality over quantity type of thing when doing these types of biological experiments. So let's look at the unique biological processes because, again, we know that these are all similar. Where do these things kind of separate and what information can we get from that? So if you look at this heat plot, it's what I've mentioned before, the unique biological processes. And again, I can't really describe without showing you how difficult the retrieval process is. As much as you want to, you will probably have many neurons that you lose. But at least getting some information during the retrieval process is really important because if you look up here, we actually do see a pretty decent clustering of what I would say is a qualitative assessment of what your electrophysiology is saying. And what I mean by that is you have these neurons that I know for a fact are torn, so these gray ones, and they don't come back significant for synaptic signaling, even though I have the electrophysiology recordings for these, and that's evidence that we actually are collecting synaptic proteins and ion channels. Alternatively, we do see that some of these neurons with absolutely no characterization were retrieved well enough to be able to contain some of these synaptic proteins. So that's really promising. So what does this additional electrophysiology tell you from these 3 neurons? Well, one, that removing them from the tissue changes them, because, again, you can't assume that the neuron that you have in the tissue is the same thing that you pull out. So the size changes, the integrity changes, and obviously, neurons don't like to get pulled out, so their health changes. And it does tell you more about the proteome. So we have a fairly good amount of consistency between these 3 neurons, at least 1,500 protein IDs present between them. And now we see something interesting that hasn't been shown before. We're able to correlate the size of the protein that we're retrieving with the size of the neuron that we're getting electrophysiologically. And interestingly enough, we still don't really see much of a correlation between the integrity of the neuron and the proteome retrieved, suggesting that if you can at least get a recording from your cell electrophysiologically, that's good enough for a quality level. Now, it'll also tell you an idea of what you should be getting. So you could, if you're an electrophysiologist, just look at a current response and be able to pinpoint, all right, we have some calcium channels, we obviously have the sodium channels, and we have strong rectification or something like that. So this recording is representative of a neuron, number 4, that was pulled out from the tissue, and honestly, it looks beautiful, there's a strong response, and you couldn't honestly not tell that it was done after the neuron was stressed out. So a lot of functional integrity is intact. Neuron number 7, it's not that great. An electrophysiologist would not use this, but again, this is a tough process. I can't perfectly retrieve these things without hurting the neurons sometimes, but you still get action potentials. They're not what we would expect kinetically, but it's still informative. And we have neuron number 6, which honestly, it was on its way out. This was a tough retrieval, and there are some markers that we were looking at during the pulling out process that were concerning, but we were still able to at least get an action potential, but not much after that, and again, it didn't really rebound to its baseline resting membrane potential. So that's great. I mean, I have more information that we can go into the BPs, but I'm an ion channel electrophysiologist, and I want to look at the ion channels first and foremost. So if you look at this binary heat plot of the ion channels, you'll see a lot of different ion channel families that we study with electrophysiology, but you'll also see, again, that electrophysiology quality and depth seems to matter for synaptic protein detection in general. Again, here I'm just showing you the different ion channels, but we've tested this for GPCRs and other signaling molecules, and the more electrophysiological characterization that you can get, the better and more coverage you'll get for some of these interesting and important targets. And again, we have some interesting separation where we have these 2 neurons that we know for a fact are low quality, but we have recordings and have characterized them. And we do have some clustering with those that do not have any electrophysiology and have some electrophysiology characterization, but we know that it's also not good. So now we're able to, hopefully in the future, do more experiments and stratify everything that we're getting rather than throwing out the data and assuming the wrong thing from bad quality data. So just to give you a quick summary of this, because it's a lot of things, the shotgun retrieval experiments at the visual retrieval level, doing no electrophysiology, that's good enough to get you protein IDs. This is good if you want to adopt the method or you want to develop some kind of assay based off of this. But if you want to do tissue electrophysiology, that'll be great for cell typing, circuit mapping, which is the bread and butter of most of neuroscience labs. But if you want real accurate reflection of your proteomics, you need to do more ePhys. If you want to get the synaptic proteins or you want to validate some of your findings, I highly suggest that you do more of these more difficult recordings. And lastly, I'm going to steal this quote from The Office. You miss 100 of the proteomes, you don't patch. And at this point, I would like to thank everyone involved in this project, John Yates for taking a chance on an electrophysiologist learning proteomics. And it was an interesting collaboration with an electrophysiology lab, as well as collaborators in industry. And of course, thanks to ASMS for allowing me the opportunity to present my first oral presentation in a field I'm very new to, proteomics. And NIH for funding all of this work, as well as the Hispanic and Latinx community in MS. And I do have a website where I blog about some of the electrophysiology experiments that I've done in the past. So if you have any questions, feel free to reach out, email. And at this point, yeah, any questions, let me know.
Hi, there's a question. First, great talk. So I also did some patch collabing. So when you do the recording, so the cells still retain the slice, right? They had a circuit. That's why I can detect the AP and also like synaptic events.
Yep.
But after you pull out, right? So though you, I guess, I did the same thing. You lose all the neurites. So basically where synapse localized. So I think the synaptic protein you detect in the mass spec, that mainly from either from the synthesized one have never been trafficked into the like a synapse. It's actually something inside the cell. Do you think that's very informative for the synaptic physiology?
I mean, at the very least, it'll tell you that you have that synaptic protein there, and what it's doing, that requires more follow-up. But that's also why it's so exciting, because you can detect the protein at the single cell level. So if you want to do some crazier double electrophysiology experiments, try to label the synapse or something like that with a chemical modification, chemoproteomics, and then measure it, and now you have a different pool of labeled and unlabeled at the single cell level. So again, that's why this experiment is so exciting. A lot of new frontiers that we can explore with better mass spec.
Okay, thanks.
Hi, Larry. That was great. Thank you very much. It's fun to zap neurons and then do proteomics on them. So my question is more about the source of the proteins. My lab has chosen to go subcellular, just measuring the proteome in the soma, because we weren't sure where the proteins would come if you pull them out from, right? These neurons are embedded in the tissue as you pull them out. How does one validate whether the proteins come from the neighboring cells, maybe cell-to-cell environment? Do you have any thoughts on this?
Yeah, so it's a complicated question, and ironically, it's a combination of take everything, good and bad, and just categorize everything. So I mentioned before that I don't have electrophysiology for some of these neurons. If you can get enough information from them, then you'll get a rough profile. The alternative is to not collect the neuron itself and just collect random stuff that you know is not neurons, and now you'll get a profile for that. But at the very least, if you do a lot of electrophysiology, maintain the gigaseal, then there's no way you could have contaminated it, because again, you have a high resistance seal, nothing is getting through that. So unless you're somehow ripping another neuron out, then it wouldn't be much contamination, but you would also know that too. So that would be an interesting experiment that you would have either a lot of luck happening.
Thank you.
Yeah.
Thank you for the presentation. I wonder if you observed any the membrane protein coverage in terms of control versus disease model, like the tissue, when you use the tissue from AD model mice, right? My guess is that my actually preliminary study shows that the membrane protein coverage was very different from in my comparative study of control versus AD mice. So I wonder if you observed the same thing.
Yeah, so it was variable. So off the top of my head, sodium channel 1.2, Dravet syndrome. So unfortunately for the sodium channels, they're tougher, lower abundance, and tough to characterize, but we see roughly 17 to 20-ish%. Some of the GABAs and the AMPA receptors, excitatory, inhibitory, we do see better coverage. It varies. DDM definitely helps, but again, we want more information on this, and if you got to do it one at a time over the course of 2 years, we'll never get there. So that's why this, for a method development perspective, is really exciting. We can start doing things to get those membrane proteins out at the single cell level. Yeah.
Larry, I have a question. Is there any merit to aspirating the cytosol and then retrieving the deflated cell? If you're really interested, it sounds like you're mostly interested in looking at ion channels and maybe membrane-associated, G protein-coupled receptors. Would that be, discard the cytosol to kind of eliminate any of those proteins that maybe were just newly expressed in the soma?
Yeah, I guess it would depend on the question that you want to ask, and that's why it's also exciting, because everybody's kind of trying a little bit of a different approach. Hopefully in the future we can get more ideas of what's going on, as somebody mentioned earlier, at the subcellular level. If you want to just see maybe more mitochondrial proteins, that might be a better approach. But if you want to maybe characterize membrane proteins, I mean, it just makes perfect sense that you have to get as much of the cell material out where they're embedded to enrich for them. But yeah, if it gets easier to do, then we'll get more in the field and new flavors of electrophysiology at the single cell level.
And then a final question. So just thinking about, this is the laborious manual process, right? You get a lot of chemical information, you get a lot of electrophysiology information, the throughput is a little bit lower. Are there any multi-electrode patches you could imagine performing? Is this something that's on the mind to try to speed up throughput?
It's definitely been on my mind. I've talked to a bunch of the instrument manufacturers, but it's a stochastic process of how you do this at an automated level. So generally, you don't work with tissues, it's dissociated cell models. And when you're trying to patch them, the robot either runs them all on the same flow channel, so you're not able to isolate them individually, or it patches them randomly, not give you any control to dispense or really get any I guess, individual manipulation of the cell movement at that level. So that's what makes it challenging, and it's easier to pull everything that you can get.
Great, thank you. All right, let's thank Larry for a great talk. And now I'd like to introduce our final speaker.